Hey everyone, this is Devashish. Welcome to Biosmosis. So this is the final part of your TIFR PYQ 2021. So in this part, we'll cover all those questions from your genetics, biochemistry, developmental biology, and all of the questions we're going to cover in this particular session. So this is the first question. Biologist Lynn Margulis is a proponent of the theory of endosymbiosis. This is a very popular theory which suggests that the mitochondria of modern eukaryotic cells evolved from bacteria living symbiotically within early eukaryotic cells. Okay, so in early eukaryotic cells, the cells engulf the bacteria, which will further formed as a mitochondria. This is the endosymbiosis theory, and it is the same thing with the chloroplast also. Which of the following is supporting evidence for this theory? So there should be some evidence, no? So mitochondria require the internal environment of the eukaryotic cell to replicate, no? Because the bacteria also replicate outside, right? It is not necessarily they require the internal environment. So this is the false statement. Mitochondria rRNA is more similar to bacterial rRNA than to eukaryotic rRNA transcribed from nuclear DNA. Okay, so rRNA will be similar. So that means there's some genetic similarity, isn't it? So it could be the answer. Mitochondria are morphologically indistinguishable from free living bacteria. No, it is not a explanation. Mitochondria are morphologically indistinguishable. So there will be no relationship, right? Mitochondria that are removed from eukaryotic cells and placed in a growth medium can generate fully functionally free living cells. No, it is not also a correct statement. So option B is the correct answer because the rRNAs are same. So they are giving some evidence that there are some similarity, right? A genetic cross was set between two heterozygous mice with agouti coat color. Okay, so agouti coat color and having the heterozygous mice. So these are the heterozygous. Agouti is dominant over normal. So this is dominant over normal. So this is the condition is given to us. So both are agouti, right? The F1 phenotypic ratio is regards to coat color. Agouti is to wild type is 2 is to 1. Okay, so 2 is to 1. So used to be, it will be 1 is to 1, right? Generally, it will be 1 is to 1 or 3 is to 1. Okay, any of the crosses. We'll give 4 results, not 3 results. So there are 3 results. That means there is some lethal gene work, isn't it? And this is a classic example of lethal gene. A possible explanation for this could be the Gauti gene converted to wild type counterpart. No. Homozygous lethality associated with the agouti locus. Yes, this is the answer, right? So when this will occur, so there will be one progeny, 1 upon 4 progeny will show this phenotype. This is a lethal phenotype, isn't it? That's why there will be 3 progenies, which should be 4 progenies. The ratio could be 3 is to 1 or 1 is to 1. It should be 3 is to 1. Okay, because this one will produce the Gauti, but this is a lethal gene. The Gauti gene is a lethal gene. That's why we will see this particular condition 2 is to 1. This is a very simple question. A group of 3 cells, the P6P, P5P, and the anchor cell. So the vulva induction question from developmental biology are clustered together in the C. elegans hypodermis, okay, the skin cells, hypodermis. Normally, the P6 cell gives divide 3 times and forms the vulva. Okay, the vulva induction, as I have already told you. So P6P will give the vulva. You find that if the P6 cell is destroyed, the P5 divides and will form the vulva. Okay, so if at all the P6 is not present, the P5P will give the vulva. It's the alternate condition. If the anchor cell destroyed, neither P6P cell nor P5P cell form the vulva. So anchor cell is the inducer, right? Anchor cell is inducing the vulva formation, isn't it? It's a very classic concept. How do you interpret that as well? The question is completely clear. The anchor cell inhibits the P6 from forming a vulva. No, this P6 is forming the vulva. Anchor cell is not inhibiting it. When the anchor cell is present, then the vulva is formed. If the anchor cell is absent, the vulva is not formed. So the anchor cell induces the vulva formation by P6 P cells. The P6P cells inhibits vulva formation by the anchor cells. No, it's not inhibiting. The P5 P cell induces valval formation by P6 P cells. No, it's not written that P5 cell is forming the vulva. So it will form the vulva if P6P is absent. If at all P6P is present, then it is not going to form the vulva. So here the P6P is present. So it is going to form the vulva. So it will not induce the vulva. So which will induce the anchor cell, obviously. And this is a classic concept also. Bacterial antibiotics can interfere with all of the following except protein synthesis, peptidoglycan synthesis, yes, reverse transcription. So guys, if at all we'll see the concept of reverse transcription, we can take a concept of the virus. Virus have genome that is RNA genome, okay? But that bacteria do not have RNA genome, they have a DNA genome, right? So none of the bacteria will inhibit this process from RNA to DNA for reverse transcriptase. There is no antibiotics which will inhibit this process, but the reverse process can occur from DNA to RNA, the transcription, which is generally inhibited by the antibiotics. So bacterial antibiotics will not inhibit the reverse transcription, right? This is a very nice question. Next. A series of beakers of water of different temperatures are placed in front of you. You place your finger in each one and select the beaker containing water of the highest temperature. How did your brain know? Nice question. The brain receives action potential of different intensities that are proportional to the temperature is a wrong statement. So guys, in an action potential, you have that concept all or none. You will generate the action potential or the action potential will not generate at all. So there will be no different intensities. So there will be no intensities. It is all or non concept. So this is a wrong statement. Action potentials are only sent when the temperature of the water exceeds certain threshold. No, because it's given that there are different temperatures. So it is a wrong statement. The number of action potential per second increases as the temperature increases. Yes, this could be the answer. If at all the temperature is more, there will be more action potential. If the temperature is little less, there will be less action potentials. This is the correct statement. Action potentials received in different part of brain when temperature are higher. No, so there is a single part of brain which sends this temperature. Okay, so C is the answer. Meiosis is used to generate gametes. When it is meiosis completed in mammalian females. So after the sperm fertilized, so during fertilization, so it is arrested the second arrest at metaphase 2. So after metaphase 2, when the sperm will enter, sperm will fertilize the ovum, then the formation of the complete division of second polar body, formation of the second polar body, and completely formation of the egg, the ovum, right? So the meiosis will complete at that time. Next. DNA was isolated from wild type and mutant type, equalized cells and separated by density gradient centrifugation. So it will have the weight, molecular weight of that particular DNA. DNA from gal strain acquired a lower position, right? So DNA from gal strain acquired a lower position. So that means gal minus, I think. It will be heavy. At all, we'll see the density gradient centrifugation, which is present in the lower. This is a heavy molecule, will stay in the lower, the light one will stay on higher. This is a very simple concept. This indicates that the mutation is caused by insertion. If at all there will be insertion, then there will be number of nucleotides will increase. Obviously, that DNA will be heavy and that will stay below in the density gradient centrifugation. Simple question. A bacterial strain has following features with regards to growth. Will grow on agar supplemented with arginine, tryptophan, and leucine. Okay, so if at all will supplement arginine, tryptophan, and leucine, we are supplementing the three things, the bacteria will grow. It is not capable of growing on media supplemented with arginine and tryptophan. That means leucine is necessary. They do not have the leucine. We have to provide the leucine from outside, from the supplemented from the media. Leucine and tryptophan. That means leucine and tryptophan. Then that means arginine. We have to supply arginine, right? However, it grows on media supplemented with arginine and leucine. Okay, if at all we're supplementing arginine and leucine, it will grow. That means tryptophan is present. So this is the combination of the bacterial strain. So leucine, arginine, tryptophan. Simple one. Next question. The pedigree is shown of three generations that show which individuals suffer from genetic disorder that is 100% penetrant. Appear after 40 years of age, generally late onset. Individuals that show the disease are either shaded circles in females, this one, and shaded males, they are the squares. Individuals that do not suffer from the disease are unshaded. These all are unshaded. Although it should be noted that the grandchildren in generation C, still below 40 years age, so obviously they will not show the disease. So might be they have the disease, they will show after the 40 years of age. Below this is a restriction fragment linked polymorphism of each individual's DNA. Individual A in generation A is labeled as AA. So AA means, so this is individual generation A, and this is the individual. Okay, right, simple. So what is the chance that the child from a union between CH and the male CI, female CH, this one and this one? Okay. So see, we will say the RFLP analysis. Okay. So here the AA crossed with AB will give rise to BA. Okay. So let me see the pattern. So see, here the pattern is like this and this. So there is a distance pattern like this and AB. So where is AB? Here. So this is like this. And you can see the upper lane is common in everyone. So we are not considering that lane, right? So next, the diseased one will have that phenotype that is a BA. What is a BA? This, okay? So this is similar with the last one. We are considering AA and that AB. Okay, okay. So the middle one is same, you can see. But none of the other ones are same. So that BA have the phenotype like this. Okay, fine, we got it. So this is the phenotype for BA. So there is a pattern in the CH. So CH, which is similar to CH is similar to AB, which is the diseased one. Like the CH will get the disease. CH will be diseased after 40 years, right? Then, so then we have that CI. So CI from a family with no history of disease. Okay. So what is the phenotype of CI? So it is same with AA. So it will have no disease, right? It will have CI, no disease. Obviously, the chances are 50-50, right? Chances are 50-50, we can get the answer. See, let us check what is the phenotype of this BB. So what is the phenotype of BB? It will be like this, B and B. So see, so this B and B and this B and A, these have a different phenotype, right? But these are B and A, right? This B and A. So this has a different phenotype. Okay. So these are the different phenotype and these are the different phenotype. But it will get disease, it will not get disease. So the child will have a 50-50 chance. They might get chances because with the union of this diseased male and non-diseased female, normal female, there will be a normal female. But diseased female and normal male will give a diseased male. So we have the diseased female over here, right? We have a female diseased female over here and the normal male. Okay, so there will be a 50-50 chance because you can see this is not a X-linked disease. Okay, so the chance will be 50-50. Okay, next question. A protein is purified from a cell extract. SDS polyacrylamide gel electrophoresis is performed for analysis of the protein using treatment with dithiothreatol. So it will act like SDS phase. It will act like it will cleave the disulfide bonds, right? It is a reducing agent and protease. The image of the gel is shown below. What can be inferred from the treatment of protein with DTT and protease? See, so this is the control. This is a normal protein. When we are having the proteases, it will get three bands. So three bands means there will be two sites for proteases. Next, if at all we are treating with DTT, it will get two bands. So DTT will break the disulfide bond, right? So there will be how many disulfide bond? A single disulfide bond. That's why we get two bands, right? Note that the red band is two lanes and protease used. Okay, the protease is zero over here. So if at all we'll see that DTT plus protease, so DTT will give three bands, and the disulfide will hit one band, right? So total how many sites? There will be two sites for protease and one site for disulfide bond. So total there will be, protein has three protease cleavable sites. No, two. The protein has two polypeptide linked by disulfide bonds. Yes. So there will be two polypeptides by R disulfide bond and two protease sites. Yes, that's why we got three bands. So this is the answer. Next one, this is the question from your general aptitude. I left the question in the first video. A heritable disease in humans starts expressing after 40 years of age. So late onset disease leads to fatality more often than not. Which of the following statements is correct prediction regarding the occurrence of disease in the population? It will be very rare as natural selection will keep the disease allele in check. No. So after 45 years, the reproductive year is crossed, right? The middle year is crossed. So after 45 years, so there will be no natural selection will work. So natural selection generally selects that alleles which are fit for reproduction. So it is a wrong statement. It will be very common as natural selection will favor the late onset. No, natural favor will not favor the late onset. Natural selection will have negligible role. Yes, because natural selection will not have any job when there is the allele is passed to next generation, the genes are passed to next generation. Over time, the disease will become milder due to the action of natural selection against the disease. No, natural selection will not act against the disease because it is causing more fatality. Okay, so the all the question I have covered from biochemistry, chemistry, and general aptitude, and from your biology. Thank you so much for watching, guys. Please do like this video if at all you like it, and please do share with your friends. All the best for examination. If at all you are liking my videos, so please do subscribe to my channel and hit the bell icon so that you could get notified after each video is published. Thank you so much, guys. See you in the next class. Bye bye.